What up, Biochem? I'm going to be doing my presentation on a paper called Poneratoxin, a neurotoxin from ant venom. This paper was by Iwa Sholachka and a bunch of other scientists whose names I can't pronounce. But the main purpose of this paper is to get the structure of a toxin from an ant. And this ant is called Paraponera clavata, which is commonly known as the bullet ant. In Venezuela, they call it the 24-hour ant. In Brazil, they call it Formigao preto, or big black ant. And many indigenous names for the ant translate to the one who wounds deeply. Now, these ants are found in Central and South America, which is lucky for us, because why it's called a bullet ant is because when you get stung by it, it's comparable to getting shot by a bullet. The difference is, though, you can kind of like treat bullet wound pain, whereas with this, you just kind of have to wait it out. And that does not sound very fun to me. So like I said, this ant has a stinger in which it uses to inject poneratoxin. And the way these toxins work is they inactivate voltage-gated sodium channels in your muscles, which will then block synapse transmission from the central nervous system. Basically, the poneratoxin attaches to these channels, your channels, and they'll keep them open so that the sodium will just constantly flow through. And that's what causes the temporary paralysis and even shaking. Other symptoms include nausea, vomiting, fever, and cardiac arrhythmia. The good news is normally they're not aggressive and they won't sting you unless you really provoke them. And the stings aren't fatal unless you have an allergy. But I have this video here by a guy named Coyote Peterson. If you've never heard of Coyote Peterson, he's a biologist who kind of educates on bugs and animals. And he's also known for like getting stung by all these different bugs and bitten by all these animals to kind of raise awareness to educate yourself about the nature, but don't be afraid of it. That's kind of his main thing. So if you're a little squeamish, I'm going to play this video. I would skip ahead maybe like a minute. And also headphone volume warning. I don't know how loud this is going to play through Zoom, but
stuck in my arm, look at that! Oh! Okay, it's off! Oh my gosh! Oh my gosh! Oh my gosh! Oh! You're right? Oh my god, it is really...
So he's okay. But yeah, you can see like the pain started absolutely immediately, and it didn't look like it was a small amount of pain either. But a little bit more info. Basically, this guy who's an entomologist named Justin Schmidt, he developed what's called the Schmidt Sting Pain Index. He just went out and got stung by like all these different bugs and scorpions and just stuff. And he developed this pain index scale, and he ranked the bullet ant sting at the top of the scale. It's got the worst pain ever, pretty much. So getting into the actual paper now, the purpose of this paper was to explore the structure and expression in insect cells of poneratoxin, as well as construct a potential bioinsecticide from it. So now I'll be focusing on the structure explanation aspect of this paper because that's kind of all I have time for. But the implications of this paper and how they can use this toxin as an insecticide was really interesting to read. I know we all have a ton of free time this week, so if you want to just open up this paper and read through it, it's pretty cool. It's only like 6 or 7 pages. But yeah, what you'll notice about the structure, which is on the right, is that it's very small. Like that's the entire protein right there. It's only 25 amino acids long, and it's wild that such a small protein can cause such significant effects in people, how this 25 residue structure can cause the worst pain you could possibly feel almost. But before we get into the actual structure of the protein, I thought it'd be a good idea to talk about how the researchers actually found the structure of the protein. And this first bullet point here is actually the entire sequence of amino acids. And this specific sequence is a synthetic poneratoxin that was synthesized, and they synthesized it by solid phase synthesis using Tbot chemistry. They then purified it by high performance liquid chromatography, and then analyzed it via multi-TOF mass spec. And their mass spec data was pretty good. They got a molecular mass of 2,756, whereas the theoretical value was very close to that. And then they also got a donation from, it turns out the same guy, the Schmidt index pain scale guy, Justin Schmidt. He donated some of his dried venom from the bullet ant. So they thanked him for that in the paper. But in the slide, I just used a lot of fancy science words, and I figured it'd be nice to kind of go through and explain what each of these means. So solid phase synthesis and Tbot chemistry. Solid phase synthesis consists of adding individual amino acids to a polypeptide one at a time, and then like washing in between each addition to keep the products pure. A temporary protecting group is placed in with the resin in order to ensure that polymerization of the amino acid doesn't occur during this process. And what that basically means is they just want to make sure that their protein that they're synthesizing is as accurate as possible. And the particular protecting group that they used was Tbot, and this stands for tert butyl oxyl carbonyl protecting group. I tried looking up and like understanding how it works, but it's a lot of OChem. But it's a very common protecting group that's used in solid phase synthesis. After that, then they used high performance liquid chromatography, which we did talk about in lecture. And basically this separates components in a liquid mixture by injecting a stream into the solution of into a column. And there are two phases, the liquid phase and the solid phase. The liquid phase is what they're shooting into the solid phase being the column, and then that will separate it out by certain properties. In this picture on the right is a chromatogram, so like that's what your data would look like. And how they specifically analyzed their data was something called multi-TOF mass spectrometry. And what that stands for is matrix-assisted laser desorption ionization, time of flight mass spectrometry. This is something I don't really understand, but the concept of it like sounds really cool. You basically take your sample and you fix it in a crystalline matrix and then bombard it with a laser. The sample is then vaporized into a vacuum, and then it's ionized without like fragmenting or decomposing it. And then the time of flight method in mass spec will determine the mass to charge ratio by measuring the time it takes for ions to reach a detector, like in the apparatus. Yeah, like I said, that blows my mind how that works, but it does. So yeah, in summary, they use solid phase synthesis, high performance liquid chromatography, and multi-TOF mass spec. And then they were able to synthesize and confirm their results of making a synthetic poneratoxin. So now that we know how they obtained the protein, I'm going to talk about how they investigated the structure of it. So the two main methods they used to actually like find out the structure of this protein was circular dichroism and nuclear magnetic resonance, or NMR. Circular dichroism is a type of absorption spectroscopy which utilizes like circularly polarized light to study biomolecules. And basically, the light will absorb differently based on the chirality of the molecules. And this will kind of give an idea of what kinds of secondary structures are present in proteins. And then for NMR, it uses a super strong magnet to analyze the molecular structure of a target molecule by observing the nuclear spins. And then it quantifies that data so that it can then be analyzed, and then you can kind of figure out more about the secondary structure. So now that we know how they synthesized and obtained both the synthetic poneratoxin and the wild-type poneratoxin, and how they analyzed the structure, it's time to look at their results. So as I said before, most of the paper was about how you can use this as a bioinsecticide and how they tested that, whereas I'm just focusing on the structure of the poneratoxin. But figure 5 is split into 4 different parts. And if you read this figure legend that I have up here, each part is like a different representation of the protein. The first part, 5A, is a sausage model of the mean structure. And how they obtained that was they averaged the 10 best structures that they got with their data. And how they defined those best structures was that they only exhibited occasional residual violations larger than 0.3 angstroms. So then they took those best structures, averaged them out, and this is what it came out to look like. The thickness of the tubes is how flexible the backbone is in those areas. And then the helical regions are found in red. So you can see there's 2 helices that's connected by a beta turn in the middle. So then figure 5B shows the hydrophobic and hydrophilic potential of the protein as a whole. And this has to do with the types of amino acids that are present at the certain parts. C shows the, it's kind of like the space filling model, but it shows the 2 helices as well as the N terminus and the beta turn in the middle. So the red helice is the N-terminal alpha helix, and then you got that green beta turn in the middle, and then that dark blue C-terminal alpha helix, and then this light blue in figure 5C is the apolar, or sorry, it's just the N-terminus. And then 5D is a stick representation of poneratoxin, and basically they're just getting rid of all of the alpha helices and showing the back, the beta turn and certain residues that are responsible for the stability of the structure. So it's just a different way of looking at it. And I'll come back to 5D later. So getting into the actual explanation of the structure, through their circular dichroism results, they found that the ideal environment mimics a lipid bilayer and has quite an acidic pH. You'll see that the pH has to be lower than 4.5. And you might wonder, how can this toxin, take its conformation and affect people if it requires such an acidic pH? And the reason that's possible is when the ant injects the toxin, it also injects a lot of formic acid. And when I was researching this, I actually found that formic acid gets its name from the Latin word for ant, which is formica. So I guess it's been known for a long time that ants contain a lot of formic acid, and this is beneficial, especially to the bullet ant, because it allows the toxin to work correctly in their prey. And in the correct environment, it will arrange itself into a sort of V shape. And like I said before, it's got those 2 alpha helices that are connected by that beta turn. So this is breaking down what part makes up the regions of the proteins. The top left, you'll see the sequence for the N-terminus alpha helix, which is apolar, and those are residues 3 through 9. And then residues 17 through 24 are the polar and charged C-terminal alpha helix at the other end of the protein. And then you got that beta turn in the middle, which is residues 10 through 16. So looking back at figure D, it specifically shows what causes the central loop stabilization. So basically, because of these residues, we have an isoleucine at 6, leucine 10, proline 15, another leucine, and then a glutamine at 17 and 18 respectively. And these residues in particular contribute to the overall structure of the protein. It allows the protein to be small, flexible, but also highly defined. It also has a highly defined hydrophobic core, which is a result of these residues in this particular order. The helix break helix organization of the poneratoxin is very similar to that found for other peptides that interact with plasma membranes, which I'm sure the researchers were happy to find out when they saw the structure of this protein for the first time. And this also makes sense because if you remember how this toxin works, it interacts with voltage-gated sodium channels, and those channels are found on, like the plasma membranes of cells and lipid bilayers. So the structure makes sense in terms of function. So getting into the discussion, they first reintroduce the toxin and they talk about how the primary sequence was obtained from the peptide mixture stored in the venom reservoir. The venom reservoir is where the bullet ant stores the toxin in its body. So they talk about that. They also had this interesting quote that I thought opened up really well for future research. The gene for this toxin has not been characterized, and nothing is known of the toxin processing during synthesis and secretion into the venom reservoir. And I'll talk about a little bit more how and venom is not a thing that is studied. So they're hoping that this will open up for opportunities to study this in the future. And then they talked about how poneratoxin is a very hydrophobic peptide of 25 amino acid residues with a charged C-terminal part. They talked about how their circular dichroism data showed that in conditions mimicking membrane surroundings, it acquires an ordered structure. And then they talked about how their NMR data shows that it has 2 alpha helices that are connected by a beta turn. So they're just kind of summarizing the results here. And then they talked about how the structure of this toxin leads to its function. So it's got the extremely hydrophobic N-terminal helix that will easily interact with uncharged lipid bilayers. And this is due to the N-terminus, which starts with a phenylalanine. And this greatly enhances the hydrophobicity index and allows for membrane penetration. And then at the other end of the V shape, you have the C-terminal helix, which is positively charged because it terminates with an arginine, and it can attach to negatively charged surfaces. And both of these aspects of the protein kind of allow it to attain and interact with its target, which is cellular membranes and then the eventual voltage-gated sodium channels. And then they opened up more into future research. They talked about how very few anttoxins have ever been described, and only one other structure of an antox and venom is known. And this other toxin is called Ectatomine, and is similar in structure to poneratoxin, but it's a bit more rigid due to disulfide bridges because Ectatomine contains cysteine, whereas if you look back at the poneratoxin structure, or the sequence I should say, there are no cysteines in poneratoxin. So they have similar function, but different structures, which is interesting. And then they talked about how using poneratoxin as a bioinsecticide and what that entails, the potential dangers that could occur to putting recombinant toxins into viruses. I didn't really talk about this, but the vector that they used for using this as an insecticide was baculovirus. So they took this toxin, put it into the viruses, and they actually found that the toxin became more effective when they were in the viruses. So they talked about like how, oh, maybe this is a bad idea. We don't know how this reacts with like stomachs of bovines, like bovine animals, because if you're spraying this toxin to kill insects, but there's leftover toxin on the plants that you're using to feed livestock, maybe we should find out if those livestock are going to be in severe pain if they eat the vegetables and stuff that it's sprayed on. But yeah, that is the paper, or at least half of the paper. These are my citations, and I hope you enjoyed.